Thank you so much for tuning into my Journal Club presentation on the study titled Environment Dominates Over Host Genetics in Shaping Human Gut Microbiota. I really appreciate you guys tuning in and listening to this. So without further ado, what does this study focus on? So the main question that these researchers were asking is, to what extent is the composition of our human gut microbiome, what part of that is determined by our genetics, the host, versus environmental factors? So what does that mean? Basically, our gut microbiome, all the bacteria that we have in our digestive system, is that determined by our genetics, our chromosomes and everything that we have inside of our body, all of our DNA, or is it by environmental factors, what we eat, our exercise, what we're breathing in? And they found that, in fact, it is the environment that's the dominant factor, and there's a very minimal influence from our genetics. So there is still some influence. Our genetics does play a role in that microbiome and its formation, but in big part, it is the environment. And that is super, super important because we know that our gut microbiome, and I'll elaborate on this in a minute, but our gut microbiome plays a huge and very important role in digestion, in our immune response, and just our overall health. So if this study has found that the environment is the dominant factor in determining how well our digestion is going, our immune response, our health, how that would be impacted, we now know, okay, perfect, the environment is affecting it. What can we do to treat and to better sustain our gut microbiome? So getting into a little bit of background here, which is really the foundation of this paper and the study, and why does this even matter? Why should we even care? Who cares about our gut microbiome? What does that have to do with anything? And I've chosen 4 studies that really elaborate on just this first slide really focuses on how it affects us and then future impacts of, okay, we know it affects us, what can we do with it? So we see that Clemente here and other researchers found that our microbiome plays a critical role, like I was mentioning previously, in physiological functions, but also psychological functions, as it says here, our mental health. And that includes digestion, our immune system, that regulation. So basically, if we're sick or if we're healthy is determined by that gut microbiome. And we also see that it's implicated in a numerous amount of diseases. So that includes obesity, diabetes, inflammatory bowel disease. All of that comes back and has an impact on how, quote unquote, happy these microbiota are, how happy our microbiome is. So it's very important that we take care of it and we realize what's affecting it for better or for worse. So going more into background or more importance, what other research has shown is that modifying these microbiomes through things like our diet, what we eat, probiotics that we can take, lifestyle changes. Are we working out? Are we not? Where are we living? So this all presents new opportunities for personalized medicine and disease prevention, which is essentially if we're changing all of these things and that therefore affects our gut microbiome, are we healthier? Is it for the better? So, and it's been shown that it is, and it kind of makes sense. It sounds redundant. Oh, I work out and therefore I'm healthier. But we really have to look at what we're putting into our bodies. How does that affect the microbiome and so on and so forth, and has this domino effect. And we also see that public health policy changes, that is super influential in reducing metabolic and autoimmune diseases. So if we can take this a step further into the system that we all live in, we can produce a change for our healthcare system for the positive, putting in these changes that kind of illuminate, okay, we know now that it's affected by our environment, our microbiome is affected and shaped by our environment. What can we do to better it? So now getting into the researchers' goals as they were going into this study. So there was 4 main things that they investigated and found data on, and they went into the study looking to see what resulted from investigating these different variables and their effects on our gut microbiome. So first and foremost, these 2 bullet points right here, they wanted to investigate, do environmental factors like diet, lifestyle, household sharing, how does all of that affect our gut microbiome rather than genetic ancestry or specific genetic variants, things like SNPs, which are specific genetic markers. How does that affect the microbiome diversity in reference to or in comparison with environmental factors? They also looked into diet and lifestyle. So that goes back to environmental factors, but they looked into specific aspects of it. So things like what people were eating, consuming, their diet, right? Their sugar levels, etc. And then lastly, if the microbiome versus the genetics, our genetics, if that affects health traits, or as they stated it, our phenotypes, right? Our phenotypes is what is expressed from our DNA, like what is seen. So some examples of that is BMI, glucose levels, our cholesterol, even our waist hip ratio, all of that, which of these is playing or has an effect on all of that. And of course, both of them do, right? So our genetics does play a role in cholesterol levels, for example, to some degree, or a waist-hip ratio, but they investigated specifically the microbiome and how it played a role in all of these factors, which is very interesting. So now getting into the methodology. So the researchers ran a various different aspects of the study, and it was all within a few separate cohorts. So the one that I will be focusing on, and which really makes the bulk of the study, is 1,046 individuals, the Israeli individuals more specifically. So the study analyzed a cohort of these of healthy Israeli individuals who provided data on the microbiome, their genetics, and lifestyle. Right. And in order to validate these findings later on, they replicated with a cohort of 836 Dutch individuals. And that was actually taken from the Lifelines Deep LLD study. So we'll talk about that a little bit more later on. And then in terms of microbiome analysis, I will get into it in a short second, but they did do 16S rRNA gene sequencing and metagenomic sequencing, which is to be expected. And that was performed to analyze the microbial species, the composition of these different types. And then lastly, there was statistical approaches to, these are just a few, linearomics models, Mantel tests, GWAS, amongst many. So we'll get into that now. So getting into more detail, we have this Israeli cohort, which is healthy participants aging from 18 to 70, and they were monitored with continuous glucose meters, CGMs, that's what they're called, for 7 days while logging activities and meals. And these participants provided stool samples that were genotyped via the Illumina OmniExpress, and analyzed through metagenomic and 16S rRNA sequencing. So it was either collected via swab, so that's 88% of them, or the OmniGeneGut, which is basically just think of it as a kit where they can collect their own stool samples and then they ship it in. So just think of it as like very similar to a COVID at-home test, for example, something that we've all familiar with. That is kind of what it is. But most of them were collected via swab, but this is just a separate method of it. And like I said, these were the two ways in which they sequence that DNA. So the Illumina NextSeq and HiSeq are DNA sequencing machines, which are or were used in the study to analyze genetic material from stool samples. So basically the way they work is by reading millions of small DNA fragments all at once, and that allows researchers to identify and study the bacteria and other microorganisms present in each sample. And essentially the difference between them is the HiSeq is designed for large-scale sequencing projects, so a lot or massive amounts of data, versus the NextSeq is a system better suited for smaller or medium-sized studies, but both help researchers get a detailed picture of the microbiome by decoding this genetic information. And then the second cohort we have here is the Lifelines Deep cohort, which included 836 individuals from 18 to 84 years old, and they're all healthy, of course. And this is a sub-cohort study within the larger Lifelines cohort, which is essentially a prospective population study that is based in the Netherlands and that focuses on collecting extensive biological data. So the researchers in this study kind of replicated theirs within this. So it's a little complicated, but that one is like the second cohort that we see there. And they did some genotyping and sequencing likewise, and they used the Illumina human cyto SNP12, immunochip arrays, and HiSeq platform, similar to the one here. And then they also did some further data processing. So we see that the Illumina human cyto SNP12 is a tool that scans a person's DNA to find small genetic differences, and that's what those SNPs are. Immunochip arrays are specialized DNA chips that are designed to study genetic variations linked to immune-related disease. So by studying these targeted genetic markers, researchers can then better understand what genetic factors are influencing these immune system disorders. So very important. And then, as I had said before, the HiSeq platform, similar to the Illumina, same thing. And then in reference to data processing, this is quality filtered reads. So they basically remove human genome DNA and they do microbial profiling via the Meta PHL AN 2.2. I hope I said that right. But essentially reads like basically all the DNA information that's put into the system is quality filtered, and the human genome is actually removed using what's called Bowtie 2. And then the microbial composition is then analyzed by the Meta 2.2 here, and that just identifies and measures bacteria in a sample of the DNA that's put into the system. So very useful. And then getting into a little bit more, once again, they have a lot of different tests that they're running for all the different things that they're studying. And it gets a little bit complicated, but bear with me as we break it all down. So the genotype quality control is essentially the filtering of SNPs, which is what we were mentioning before. And that's based on, they're taking out SNPs based on missingness, the Hardy-Weinberg equilibrium, allele frequency, batch effects, all of that. And they ended up with around 500,000 SNPs. So it's kind of like filtering out, think of it as like picking out the bad, leaving in the good, quote unquote, of what researchers want to keep in. And these genotypes were prepared using reference panels from the 1,000 Genomes dataset and the Ashkenazi Jewish individuals. So basically they prepared this using what we already know in the genomes dataset of the population, which we know is like the Jewish population. So they used that specific pre-made dataset to make that, and ended up yielding around 5 million SNPs, which is what they were using. And then they did microbial pre-processing, which is removing low quality reads and filtering out human DNA, which is what we were mentioning before. Basically just like quality control, and removing what they don't want in there, and basically studying like only the genetic composition versus the microbiome composition. So what percentages we have there. Then we have the fasting glucose and the glycemic status. So this is derived from the continuous glucose monitor, which is what they were using, the individuals or participants. And it's taken from that data using HbA1c, fasting glucose, and glucose response metrics. So it uses the median glucose level within 30 minutes of waking up. So that's like the fasting glucose. And the glycemic status was determined based on the following, what I just said. And then lactose consumption was also studied, and played a role in a different part of their study, a different gene specifically. And then genetic kinship and relatedness. Once again, there's like many branches of this, and this is another one. So they assessed this using, so genetic kinship is just think of like, okay, how related are you to this person and how that relates to your microbiome. And they assessed that using PC-Relate and PC-Air. So PC-Relate is a tool that estimates how closely people are related based on their genetic data, even if they have mixed ancestry. And this helps identify relatives while accounting for population differences. So, and then we have PC-Air, which is a tool that finds genetic patterns within a group of people while avoiding bias from relatives. So it's just basically made out to process that bias and see, okay, you say you're related and have this line and this genetics, let's just double check that and make sure that that's all true, because we can't be comparing two people that we think have the same DNA, have the same ancestry, when in reality they don't. So that's just like quality control, making sure that that's all accurate. And just kinship coefficients are going to determine that relatedness. And then finally, we have ancestry and microbiome associations. So for that aspect of the study, they used Mantle tests, that's basically used to check if there was a connection between a person's genes and their gut bacteria. So depending on other factors that might affect the results, which are called covarieties, so that includes age or diet, based on that, different methods were used to measure the strength of this connection. So basically, Mantle test is like the overall one. But if we have other stuff that come into effect, like covarieties, then we would use the Spearman correlation. Then we would use the Pearson partial Mantle test. So that's used when extra factors are included, and that's used to measure the relationship between two things while removing the effect of those influencing factors, those external factors. And then the Spearman correlation is if no extra factors are considered, then that's used, and that measures how strongly two things are related by ranking them. So if it basically checks if one thing increases, will the other decrease, or vice versa, like decreasing. Awesome. So now let's get into our results. What does all of this tell us? What are we learning from it? So here we have this very, very big, this is a first figure. So do not fret. I'm going to break this up into A, B, C, and D. We're going to break down what each of the graphs tells us. But overall, basically all of this concludes into one big idea, which is that genetic ancestry is not significantly associated with microbiome composition. And we're going to see that right now going into each of these graphs. So first and foremost, we see here that we have principal component 2, principal component 1, that's our X and Y, and we have this all grouped together. If we actually go back, I actually have this all up here, what these like dots mean, the coloring. So get into here, that's what all these mean, the coloring. So genetic patterns, which is what principal components are strongly linked to self-reported ancestry. And that includes groups as Afro-Gandhi, North African, etc. All that was stated there. And we see that by a very, very low p-value, right? So that makes sense, right? Genetic patterns are linked to what we say our ancestry goes back to. So that's just kind of confirming that makes sense, right? And then if we look into this other graph right here, we see NS, right? Which that means non-significant. We're going to see that in some other figures here. So it's important that we know that NS is non-significant. And here, once again, we have PCO1, which is principal component 1, principal component 2, and now it's being compared in reference to the microbiome. So we see that when analyzing the microbiome composition using that same method, there is no significant link to ancestry found. We have a p-value that's greater than 0.08, so it's not significant. So we see that ancestry has nothing to do, all of those genes passed on has nothing to do with the bacteria that we have in our stomach. So we see here, among 582 individuals, which was particularly studied in this graph, with a single ancestry background, the overall bacterial makeup at the phylum level, after normalizing data, after running all that through, it showed no clear connection to ancestry, right? So non-significant, and we had a p-value that was greater than 0.05. So once again, we're looking at all of these different phylums, right? And we're comparing that with these different lineages or ancestries, and we see that it's not significant. The connection between them does not correlate. There is no connection. And we're comparing that same thing with grandparental ancestry, all different types of ancestry, and it's just not showing any significant association, p-values of greater than 0.05, greater than 0.06. So all of that was run. And then these are just a few examples of like the phylums that they ran and like saw connection with. But once again, there was none. So there's no strong link between the top 5 genetic patterns of a person and the top 5 diversity patterns of their gut microbiome. So there's no correlation between them. And then now getting into these box plots, right? So here we have microbiome dissimilarity, and I'll explain what that is now. And then we see once again all of these like ancestries, and the most important thing, non-significant, right? So kind of going back to establishing the same thing that we were saying before. So these are box plots of microbiome differences, and that's basically called the Bray-Curtis dissimilarities between pairs of individuals who also showed no relationship between microbiome similarity and ancestry. So this was true when we were considering different levels of shared industry, of shared ancestry among 946 individuals. That's what was the population that was used here. And they had mixed backgrounds. But once again, this is just all reiterating the same thing, which is right here, which is that there's no significant association with microbiome composition. And we see P levels of greater than 0.73. And just essentially reiterating that the findings suggest that ancestry does not significantly shape the microbiome composition. So now getting into another aspect, another finding that they found. So genetic kinship is both of these are showing that genetic kinship is weakly associated with the microbiome. So once again, non-significant. And what are we looking here? So genetic kinship and then microbiome dissimilarity. So this scatter plot shows the relationship between this x-axis, our genetic similarity, right? Like our kinship, how related we are, and microbiome differences, which is right here on the y. And the results show that there's no connection between genetics and microbiome similarity. We get a p-value of 0.59, so clearly not significant. And then this R value down here, we see this line. It's just all of this is demonstrating that there is no significant connection, right? And then here we have another analysis. So we see that this is in the light green, the bars show the number of bacteria that are considered heritable, while the dark bar shows the total abundance of that. So this graph right here, let me backtrack a little bit. This is basically the researchers got a study that studied 2,252 twins. So there was a specific number, but basically it was studying a group of twins, and it was seeing how their gut bacteria traits were inherited within these traits, within these twins, excuse me. So this is not an original findings there. They took a study and they basically took their information, took their test, and they ran it again. And they were like, okay, let's see if the percentage that they claim, the previous study claimed, just for example, I don't know, 15%, right? That there's a connection between the twins and whatever microbiome, whatever component of their microbiomes, the composition of it. And they ran it again, and they basically saw a 1.9% heritability. So that basically means that they took this study, they were like, okay, let's run it again. Let's take all their information, see what they find, because it's twins, they share the same DNA, and see if that's affecting their gut microbiome. And they found that only 1.9% of it was heritable and was found in that microbiome abundance, right? So this is what's inherited. This is what our gut microbiome is as a whole. So I know that this is not actually like part of like their big study and what they found, but I did think it was super important considering that they took that study and they found such a small heritability amount, which kind of feeds back into, and that's why they included it in their paper, kind of feeds back into the total and overarching idea that this is not related at all. And even though there's a very small amount, which is still worthy of investigating, and we'll get into that later, it's not significant at all. So another finding that we have here is this shows limited evidence for microbiome associations with specific SNPs. So this right here is a Manhattan plot, and this displays the lowest p-value for each tested SNP, which SNPs, once again, are genetic variants, right? To see if it's linked to all these different types of gut bacteria. So these associations were analyzed using data from the X amount of individuals that they were studying, and the dashed lines on this plot mark right here, these show statistical significant levels. So the top line shows the stricter thresholds after adjusting for testing multiple bacteria types, and then the bottom line shows the unadjusted threshold, right? So this right here kind of shows everything's below it, right? So it's not meeting that threshold. So there's very, very limited evidence. You still see a few dots here, but this is p-value. We still see a few dots here, but there's very, very limited evidence for all of that bacteria that they were studying, all of those different gut bacteria that they were running, really show no association with genetic variants, SNPs, which are like these markers. Like think of it as like a yellow marker in your DNA that's telling you, hey, I'm specific to Sally, I'm found here. And then you see that in Sally's daughter, and so on and so forth. It's kind of like a marker, like saying, okay, this is standing out here, and that is not associated with our microbiome, right? It's not like saying like, okay, because this X person has that SNP, then it's going to affect the presence of X amount of bacteria. And then here we have another finding, once again, establishing very same thing. So this is a quantile, which is called a QQ plot. Kind of fun to say. And this shows that only 2 genetic variants, SNPs, have a strong link to overall microbiome diversity. So going back to what we were saying in the previous slide, the previous graph, what does this have to do with it? So this shows us that right here, we have like a very, very thin line of like, okay, it's kind of close. We can kind of see it, but it's only because they found that 2 of these SNPs have a very strong link to it. So I decided to include this because I did think it was very important in the sense that we should investigate those 2 SNPs and what effect they have on our microbiome, on the gut microbiome, because even though it's very little, it's still a branch that needs to be investigated and something needs to be looked into because it still does affect, and those 2 very specific SNPs do affect our gut microbiome. So we need to look into that and kind of see that, and that's one aspect that they included to show those specific ones. But outside of that, the majority of SNPs, once again, don't have that connection. So now getting into another graph. This is a lot, so I'm going to break this down, but essentially this all comes down to the gut microbiome use in inferring a significant fraction of the variants or our variants of several human phenotypes. So how can we use this, our gut microbiome, to see what phenotypes we will be showing? So this is huge and a very important part of it. So looking into the first one here, A. So if we look at this, we see that a measure called B squared, so just kind of break that down, is an estimated, or it estimates more accurately than genetic heritability when analyzing gut microbiome data. So B squared represents the proportion of variance in a trait that can be explained by the microbiome. So it's kind of similar to H squared, which is heritability, and that measures its genetic contribution to a trait. So we're using B squared to quantify the microbiome's influence on different human traits. So BMI, cholesterol levels, glucose levels, etc. So a higher B squared value means that the microbiome plays a stronger role in determining that trait, right? So here, this is our like microbiome association, that's like our B squared, and then this is genetic heritability, that's the H squared. So we're really focusing on this, and our Y right here, the average 95%, this is a confidence interval, and a narrower confidence interval means that we have more precise estimates. So this shows us that our B squared, which is this right here, is a better estimate or better predictors of what we're going to go into now, which are those traits. So here we have a bunch of those traits. So I just want to touch on this briefly because we'll look into it more now. But we see, of course, in the Israeli cohort, and then this was the replication, but really focusing on this is that microbiome association index. This is the percent of how the microbiome, our gut, our bacteria, is affecting our phenotypes. So essentially 36% of that association is affecting our cholesterol, or HDL, right? Our lactose consumption, waist circumference, hip circumference, etc., glycemic status, all of these things, which are super important to honor. I mean, like it's even very small, but our height, all this bacteria is affecting all of this, and in pretty significant amounts, like 30%, 25%. So that's kind of shows us it's worthy to look into and to investigate, like, okay, how can we, once again, quote unquote, make this bacteria happy? How can we feed our microbiome positively to affect these factors all in a positive way? And this is another way to look at it. But essentially, they broke all of this down. And what they found was that 8 out of the 12 traits were strongly linked to the microbiome. And all 12 were in some sense linked, but 8 of them were strongly linked. And that's after considering age, gender, diet, genetics, all these extra things. That's after considering all of that. And still 8 out of 12 of them were strongly linked. And the microbiome influenced these traits on the following levels, right? Which is what we're looking at here. And additionally, if we're adding microbiome data, that significantly improve the prediction accuracy for 10 out of 12 traits. So essentially here, what we're seeing is this is before, this is after. We add in the microbiome data, like we add in, okay, once again, bringing in Sally as our example, she has this composition of this bacteria, 25% of this bacteria, 50% of this bacteria, etc. Once we put that in to predicting what her BMI, cholesterol, all of that, it showed us a better prediction of her genetic risk scores, which is called a PRS, right? So putting that into it, it showed us that we were better able to predict all of these traits, all these factors when we took into account the gut microbiome, which is huge. And then finishing off here, we're getting close to the end, but once again, this like latter second part is super important because we can see how it's relatable to us. Like we all have a BMI, we all have these traits that affect us. So if we're looking at our gut microbiome, how can that affect those traits. So here we're looking at now getting into the household. So individuals who share a household at present or have shared one in the past have significantly similar microbiomes. So close family members or just people who have lived together have more similar gut bacteria and bacterial genes that those that are unrelated or do not share a household, and they have AP level of 0.01. So this shows us phylum level, species level, bacterial genes level. So they're just all different here. But then if we're going to look at first degree relatives, second degree relatives, and then shared households, right? We see this little bar right here is in terms of microbiome dissimilarity super low. Whereas this one, or like these two, where it comes to that data is high. So we see that this comparison of first degree relatives, so that's just like parents, siblings, etc., who have lived together at some point. If they've lived together at some point, then that will lower, right? Then we have distant relatives who have probably never lived together. It's the highest, right? This is just an example of phylum level, but it's higher. And then unrelated people who are currently sharing a household, once again, lowers, right? The dissimilarity between their microbiomes. And then all other individuals included here. So we see that the significant levels are very, very low. When you live together, you're eating the same things, you're breathing the same air, you're going to have similar microbiomes, which once again tells us our environment is affecting that. And then getting into the last graph we have here, which is arguably, to me, the most interesting, but I've been saying everything is the most interesting, but this shows us that the gut microbiome is significantly associated with multiple environmental factors. So this shows that the variation in the microbiome's diversity can be explained by different environmental factors. So this analysis was done on all those individuals and the individuals in the study, and these numbers in the parentheses show how many factors like diet, lifestyle, etc., are included in each category. And the amount of variation explained depends both on how the category influences the microbiome and how many factors are included in that category. And that's all based on the questionnaire used in the study. So essentially, if we're looking at what vegetables you ate, right, and the explained variance, this is showing us like, okay, is it more dissimilar? Is it less dissimilar? Like the vegetables we're eating, the fruits we're eating, all of this stuff, beverages, this is just an example, but if you look at all of this is affecting how varied or what gut microbiota we have, what bacteria we have in there. So that's super important to consider because it makes us think twice about, what our diet is or what vegetables we're consuming, what meat products, all of these things. And there's other things like age and gender, that you can't really help. But outside of that, these things are affecting us and our environment is affecting us. So what environment should we place ourselves in? What should we put into our bodies? And that'll lead me further on into the discussion. So we see that the microbiome plays a huge role in many aspects of our well-being, digestion, immune function, even our mental health. So knowing what it affects can help us develop better treatments for diseases and improve overall health. So we see the impact here, right? It shapes more than the environment is shaping more than the genetics is. And we do have to look at some limitations of the study. So it definitely needs for a larger, more diverse cohorts. It was like 1,000 something of Israeli individuals. So definitely need more diversity and just to be expanded. So that would reveal a lot more. And then also from here, we just need further research on how these environmental factors impact the microbiome. So very briefly in that last graph, we investigated that, but it was very brief. We need more research on it. We need more details as to specifically what environmental factors affect the gut microbiome and like how, and then some broader implications are that this supports microbiome-based health interventions, looking into that aspect of it and how we can change healthcare based on this information. And the antibiotics that we prescribe, the rate at which we prescribe antibiotics, we should think twice about that because of information like this. And then we see the potential applications in obesity, diabetes, and metabolic disorders. And all of this kind of just takes us into like a bigger realm of how can we treat others with this information that we have. And now essentially wrapping up some main takeaways is that environmental factors do dominate microbiome composition. And we see that there's a minimal genetic influence. There's still some, it's important to look into, like we mentioned, those two SNPs that are strongly associated. But outside of that, we really do need to focus on environmental factors and how specifically is it that they affect it. And we see that household sharing matters. So living with these other people is more important than what our microbiome, or it affects more importantly what our microbiome is composed of than who we're related to. And we see things that some future steps that we can take is targeting lifestyle, dietary changes, how that would, or doing research on how targeting these things can affect our microbiota. Looking into diets, drug use, things like BMI, waist circumference, how looking into all of that can basically lead us into personalized medicine. So precision medicine, right? So how can we cater treatment to you based on what you have told us and and what you've described in your microbiome. So that leads me into some questions for further discussion. I won't read them, but I do look forward to seeing what you guys think about these questions that I'm posing. I'm also curious and have some ideas and thoughts to share. But for now, I will wrap this up. These are my references, and thank you all so much for your time.